Thank you all. Thank you all for joining the Immunology Technology Showcase, sponsored by the Colton Center for Autoimmunity at Yale. I'm Joe Kraft. I'm the scientific director. I'll introduce myself along with the director on the next slide. But we welcome you all and very much appreciate you coming so early in the morning. And we look forward to hearing some excellent scientific presentations. So the goal of the Colton Center is really to provide slide advance. Thank you. Can you show up there? Yes. Thank you. We're still. I'll let you. As a scientist, I'm used to drawing on the blackboard. We do talks. It's a little hard in this room, so maybe next time I talk. So, but thank you all for coming. So I'm Joe Kraft. I introduced myself. This session has been organized. Really, the brains and the muscle behind the operation is Makoto Yoshioka, who is the director of the Colton Center. Makoto's been with us for about a year, and he's really, I think, enhanced the role of our center in trying to develop translational work in autoimmunity and allergic diseases from Yale throughout the larger community. So thank you all, and kudos to Makoto for putting this together. So the Colton Center was funded by a generous gift from Judith and Stuart Colton about 5 years ago at Yale. We're a member of a larger Colton consortium. You'll hear speakers from all four centers today. NYU was the first Colton Center. Yale, Penn, and now Tel Aviv. And we're all interested in the same thing, which is to take work out of academic labs who are interested in immunological, autoimmune, allergic diseases and trying to translate that into the commercial space. And that's what we'll talk about today. And you'll hear presenters from all four institutions. Because Tel Aviv and even Philadelphia is a bit of a ways away, we'll also hear presentations by Zoom. We'll start with a few presentations in person, and then we'll go to Zoom later on in the session. So this is our mission, and really we want to facilitate the development of novel diagnostics, therapeutics, and platform technologies, and that's what you'll hear about today. It's really a broad group of autoimmune diseases. I'm not going to go through these in detail. You'll hear about several of these today, but it's really to focus in several areas: disease mechanisms, basic biology, but basic biology as it can be applied into the therapeutic space, diagnostics, therapeutics, and platform technologies. We're doing all those things. We recently had our center meeting at Yale. We heard from 36 applications from Yale investigators. We'll fund a good number of those. Those funding decisions are being made now, and we have proposals that will be supported in all these areas, and including artificial intelligence and how that can be better used for therapeutic design. We've had some successes. You'll hear about one of those today, Plethora, from Ian Odell, who's a member of our faculty who works with Richard Flavell, as well as other therapeutic successes, even after only been in existence now for 4 years, going on 5. So I think we've had some good success, and I think with Makoto joining us, we'll have more going into the future. We have an advisory panel. Thank you all for coming. Thank you for taking the time to do this. They'll help us in terms of asking questions, thinking about proposals, and providing input to investigators going forward. So thank you all. I won't go through all the names, but they're shown for you here. This is our symposium speakers. We'll go through those today. We'll ask each symposium speaker to introduce him or herself as we go along. We'll try to stay on schedule. Everyone has 5 minutes, slides predetermined in terms of formatting, so we can stay on time. And then there'll be 5 minutes for discussion and questions. So it'll be brief, but these are really pitches for investigators to present their ideas and summary of those ideas to you all. So with that, I will stop and we'll go to our first presentation. If I can get some hands with that. This will be from Bill Damsky at Yale. And I'll ask everyone, again, all speakers to introduce themselves before they get started. Thank you all.
Perfect, yeah. All right, good morning, everyone. So you actually get two of us for the price of one. My name is Bill Damsky. I'm a dermatologist at Yale.
I'm Jeff Cohen. I'm another one of the dermatologists at Yale.
We're going to talk to you today about the DermSite platform. Here's our team, two of us. We have an incoming dermatology resident working with us as well. I want to spend some time focusing on what it's like to be a dermatology patient with inflammatory skin disease. We're going to take the example of psoriasis. We're also going to talk about eczema in this talk. Imagine yourself developing one of these disorders, and it may even take you weeks or months to get in to see a dermatologist. Your skin may be red and scaly, you may be itchy. You're seen by dermatology, and really the standard practice now is to empirically implement a biologic therapy. It may take weeks or months to even get that medication approved. Even after the medication is approved, some patients don't respond to therapy. Thankfully, most patients do, or many patients do, but others don't. And so estimates in atopic dermatitis and psoriasis, atopic dermatitis being more or less equivalent to eczema, suggest that a large number of patients don't have a great response to their first therapy, and they actually switch medications. Really, this creates an opportunity and an unmet need. This trial and error method is inefficient, wastes time, money, causes frustration from patients. Here are some estimates about the number of dollars that this costs the healthcare system each year. The work of us and others have shown that patients with psoriasis and with atopic dermatitis or eczema actually have a lot of heterogeneity. We personally think, and there's a lot of evidence to support, that this immunologic heterogeneity relates to or correlates with unpredictable treatment responses. So one patient with psoriasis treated with a medication that has one mechanism of action may respond very well, another patient may not. And really, we think that it's this underlying immunologic heterogeneity that underscores this. And so just to sort of reiterate the opportunity here, we're initially focusing on psoriasis and atopic dermatitis because they're very common. There's a number of FDA-approved biologics with distinct mechanism of actions. And really the opportunity is to match that individual patient with that best medicine for them. And I'll turn it over to Dr. Colton to explain more.
So this is our solution to this problem. We've created the DermSite platform, which is a painless and rapid approach to personalized medicine for inflammatory skin disease. The way that this works, briefly, is a drop of detergent-based gel is placed onto lesional skin. We then use a cotton tip applicator type device, almost like a Q-tip, to rub for about 45 seconds, the size of a pencil eraser, to create what I like to call a cytokine soup. This has a lot of the important proteins that are going to tell us how someone's skin is going to react to a certain medication and may help us match that patient with the correct medication. We then take this cytokine soup, solubilize it, and can do one of two things with it. The first is a rapid readout, think rapid strep test, rapid pregnancy test, home COVID test, where we can identify which cytokines are present in an individual patient's sample and try to match treatment to that. The second output is a more advanced send-out type test where you send it to us in a lab. We do more in-depth analysis of the cytokines and return that to you. This is something that dermatologists say they would use, say they need, say that they would choose treatment based on this. And our approach has actually been able to show who's going to respond to dupilumab, a common medication for eczema, and who will not. There are a few other competitors or other companies in this space. The first is Mandira Health. They do a lot of what we do, except they are mRNA-based. We are protein-based. We think being protein-based is a little bit better because it goes right to the target of the treatment, and also because we're able to sort of change what we're looking for very rapidly as new medications come out, and more are coming out each day. Our approach is also totally painless, does not puncture the skin at all, no scar, very easy for patients to tolerate. We've done this on hundreds of people, and they've all tolerated it very well. And we feel that our costs will also be competitive as compared to some of the other companies that are out there. We're now working with Rowena Johnson, who's a regulatory affairs consultant, to talk about Q submission for the FDA. So here's our Gantt chart of sort of where we've been and where we're going. We've done some foundational work using a different approach to show that this will work. We've looked at some of what we've described so far today with a few hundred samples, and we're collecting more. Now we're working with an engineering firm to perfect our readout card, the ELISA test. We're going to continue to perfect our algorithm and bring this forward and use it in sort of clinical trials into 2026. Thank you very much for your attention, and we're happy to take questions maybe towards the end. Shot clock. Yeah.
Really interesting. Is the issue distinguishing between psoriasis and eczema, or is it distinguishing between what medicines will work best for psoriasis for that patient?
We could not have planted a better question. Thank you for asking that. We are able to do both. So we can distinguish between psoriasis and eczema based on sort of the key hallmark cytokines for one condition versus another, which are different. However, what we're really trying to do with this, and the question that we're trying to answer, the problem we're trying to solve, is personalizing treatment. We have a lot of different choices that go at different targets, and this can be used to sort of show that patient A would do better with medication X, whereas patient B would do better with medication Y, to try to simplify that process, save time, money, and help patients do better.
And is that to be developed, or is that already discovered?
That's already discovered. What we're doing is continuing to sort of perfect that and add targets as more medications come out and so forth.
In the whodunit of psoriasis, whodunit? Is it dendritic cells? Is it T cells? And so to direct these kinds of therapies with your technology, whodunit?
Yeah, so both dendritic cells and T cells are involved. Dendritic cells are involved early, and then T cells are really the ones that we think are central to this, particularly the Th17 cells in psoriasis and Th2 cells in atopic dermatitis. And so most of the cytokines that we're observing here and that we're looking at and that the medications are targeted to come from those T cells. And that's really where the action is therapeutically.
Could you say a little more about what you've done to try to really understand the predictive value of the assay, even using people who are not getting therapy driven by your test, but prospectively looking at that? Because that could be an important scientific and value creation question.
Yeah, thanks for that question. So I guess I would say that a lot of this work was done retrospectively. We're trying to enroll folks prospectively. We're not using the biomarker profiles to choose treatment at this stage, but we're more sort of collecting the biomarker profiles. Patients go on therapies as what's typical of standard of practice and sort of trying to understand how they do based on their biomarker profile. I would say in atopic dermatitis, we have some really exciting biomarkers that correlate with response to dupilumab or Dupixent, which is a common eczema treatment that targets IL-4 receptor alpha. So I'd say our biomarkers in atopic dermatitis or eczema are pretty advanced. In psoriasis, we're still sort of working on them, but we have also some exciting candidates. Please.
Yeah, can you speak to sample stability? I know you mentioned Medera, they have their patch, right? And you guys are swabbing and then putting into some kind of solution. Can you speak to, I guess, survival of the sample across transit, things like that?
Yeah, that's a good question. So we collect our, as Dr. Cohen explained, our samples using a detergent. It's a non-denaturing detergent, so the proteins retain their conformation. It's put into an aqueous solution PBS that has protease and phosphatase inhibitors. We know it's stable for about a week at 4 degrees. We tend to store these in long term at -20 or -80 eventually once they go into the archive. We haven't gone beyond that to determine stability, but I think that would be pretty easy. We could do that with a mouse or something like that with a model of inflammatory skin disease and store it sequentially over time and just sort of understand how signal degrades or may not degrade over time.
Thank you, Bill. Thank you, Jim, very much.
Thank you.
Thank you.
Good morning, everyone. My name is Chris Pittenger. I'm a professor of psychiatry and deputy chair of the Department of Psychiatry here at Yale. And I'm going to share some work done with the support of the Colton Center on new markers and treatments for children with obsessive compulsive disorder. Maybe not the first autoimmune disease to come to your mind, but I'm hoping to convince you that there's a really important clinical need and opportunity here. I want to thank the Colton Center for this funding as well as for the invitation to present here today. Whoops. Here's my team. John Chu is an extraordinarily experienced research scientist who works in my laboratory. He's got the best hands I've ever worked with in my career and has really driven this work forward. Kevin O'Connor is a very talented immunobiologist who's complementing my own experience as a neuroscientist and a psychiatrist to move this project forward in exciting ways. I'm going to figure this out. Okay. So obsessive compulsive disorder is underappreciated, but an enormous source of suffering in our country and in the world. It affects about 1 in 40 people, including around 2 to 3% of children. And in children, it can cause a lot of suffering, but also it can disrupt families and it can disrupt their development and ability to move through to live productive lives. Some cases of OCD happen overnight. Like, it's amazing. Like, a parent will say, my kid was normal yesterday. This morning, they can't stop washing their hands or they can't stop checking. The pure cases are incredibly dramatic. Then there are other cases that are less pure. I'm going to get back to that. It's been hypothesized that these cases may be related to an autoimmune etiology, specifically that there's an infection which leads in a genetically susceptible individual to an antibody response, which leads to brain inflammation, which then leads to behavioral symptoms. And that's really important because if there's an immune etiology to some of our psychiatric disease, it requires a totally different type of treatment. Being able to understand this and being able to identify it and being able to target treatments is a huge clinical need. The frequency of this isn't known. The pure clinical cases where it's crystal clear something's going on are fairly rare, but it's been estimated that up to 25% of cases of obsessive compulsive disorder in children might be associated with such an etiology. We need better tests to ask that question. Unfortunately, this was first described in the mid-90s, but 2 decades of work have failed to crack the problem of what's going on immunologically. People have looked for the antibodies, they've looked for the pathophysiology, lots of hints, suggestions that something's going on, but no one has been able to pin down the etiology in a sufficiently specific way to develop a diagnostic or a therapeutic. So we've came into this space about 10 years ago, and we've made some real progress in this space, which I'm going to share just a little bit of with you. And we think we're on the cusp of being able to develop specific diagnostic tools, and then a little bit farther down the line, perhaps targeted therapeutics. Okay, so what's the clinical need? I've already mentioned some of this. OCD, most people in the room are probably surprised by the numbers, but OCD is really common. 2% of children, that's like 1 million children in the country, many underdiagnosed, many undertreated. And if it's 25% of those have an autoimmune etiology, that's a huge target. Even if it's a smaller target, the clinical need is enormous. The cost of treating these children is substantial. It's been estimated in one study as $11,000. That's a mix of easy treatment, where it fluctuates on its own, and very complicated invasive treatment. Other psychiatric disorders that might have an autoimmune etiology are incredibly common, and we won't know until we get the right tests. There is one panel, and this would be the competition for the diagnostic technology that I'm suggesting. There's one immune panel that is used to treat these children. Unfortunately, it doesn't work. It's based on findings 15 years ago that haven't replicated. It's not covered by insurance because it hasn't been proven to have predictive value. Parents pay out of pocket $1,000 or more for this panel, and ultimately, it usually doesn't inform treatment. Okay, so here's what we've done. We looked for the antibodies in these children, and rather looking in the first pass for the molecules that they bind to, because I'm a neurobiologist, we looked for the neurons that they bind to. And lo and behold, after some stumbling around and a bunch of screening, we found them. What's shown here in a slice of human brain tissue is how serum from a child with this disorder called PANDAS, that was explained in a previous slide, pediatric autoimmune neuropsychiatric disorder associated with streptococcus, PANDAS. So the antibodies from that child stick very specifically to these neurons. They're called the cholinergic interneurons in the striatum. I could go on about them for 2 hours because like my favorite cells in the brain, but they won't give me that time today. But the antibodies stick to those neurons. And what you see here is antibodies sticking to those neurons at a background level in control samples and elevated in PANDAS samples. Not in every child, not good enough signal to noise for a diagnostic assay, but a clue into the pathophysiology. Furthermore, when we treat the children, that binding goes down. That's what's shown on the right. Every single child, when treated with an immune modulating treatment, IVIG, they got better. We picked children who got better, and the binding went down. Okay, so now what we've done is we've begun to identify the specific molecules. Now let me know the cells. We're identifying the specific molecules using a screening technology called REAP with a colleague named Aaron Ring, and we've started to find several. This is the first one. Those are the wrong graphs. All right, I don't know why that's showing that graph. Something about the cropping. Anyway, you can see, it's not as impressive as in the graph I wanted, but you can see that there's a statistically elevated binding. The important thing is this is a specific antibody, and it's a high throughput, cheap high signal to noise ELISA assays. So this is the direction we're going as we find the specific molecules. All right, so what are the opportunities? We're finding more and more of these antibodies. We have one that's really well developed and several others that we are following up on now. Competition is limited and not of clinical value that's been proven. So the future potential is, so we believe that we're close to being able to develop diagnostic assays based on what we're finding in the lab. And that technology for that's pretty straightforward. The key is knowing what to look for. Down the line, we imagine that this could lead to personalized medicine, that if we know the specific antibody in an individual that is leading to their pathophysiology, that we can do a specific treatment rather than these sledgehammer immune modulating treatments like IVIG that are used currently. There are some challenges. We're not finding one antibody in every kid. That was the dream, that 80% of our kids would have the same antibody. We're not finding that. It's going to be heterogeneous. It's going to be a bunch. And we have not yet, to anticipate a question, we have not yet proven the predictive value of these tests. That is the next step. Milestones, we've had funding from the NIH. We've published some of this. We're developing our first antibody target. We have filed a patent on the first set of antibodies, and we will continue up as we discover new ones. And the next step is to further characterize, to do a prospective trial, and then ultimately, though this is a couple years down the line, to move towards specific antibody targeting interventions. I don't have a clock here, so I don't know how I am on time, but I'm done, and I thank you all for your attention.
So IVIG works in this the children, and what's the clinical endpoint if you anticipate this being a clinical trial?
Sure. IVIG works some of the time. Clinically, it looks like it's about 50% of the time. The controlled trials have been very small, and they have mostly failed, I think, because they are too small. So in the children I showed you, those actually come from a trial that didn't hit its endpoint. We picked the kids who responded because we figured those would be the kids who would be most likely to have antibody-mediated pathology. Part of the problem is that these clinical cohorts of children are very heterogeneous. And so again, once we have assays, one thing we'll be able to do is move towards clinical care, and we hope that they'll have predictive value, but they'll also be enormously valuable for the research because they'll enable us to recruit more homogeneous cohorts for our treatment trials. You asked about the clinical endpoint. It's based on behavioral and clinical symptoms. So there's a standard measure of OCD symptoms developed here at Yale called the Yale Brown Obsessive Compulsive Scale. It's got excellent reliability. I mean, it's not a blood test. We have no blood tests for these things, but it's got excellent reliability. There are also other modes. These kids usually don't have just OCD. They also have separation anxiety, bedwetting, motor symptoms, deterioration of handwriting. All of these things can be followed in an individual case, and success entails enduring improvement in all of those symptoms. As we understand the immunology better, of course, we can also look at immunological endpoints, though those, in my mind as a clinician, are surrogate endpoints, which ultimately are value insofar as they correspond to or predict the clinical outcomes.
Thank you.
I want to add on to that question. How quickly does IVIG work in those at risk?
Weeks. It's variable, like 2 to 4 weeks. The endpoint in these trials is usually 6 weeks. Yep.
I know that you said that the validation is ongoing, but
And I knew you were going to ask that question.
sorry. Could you say a little bit about when you find the antibody, whether retrospectively it correlates to that acute onset, which is kind of part of the hypothesis, and also do you know whether kids who don't respond or don't get better, does their antibody stay up or does it go down?
Two really excellent questions. The assays that we've used up to this point are very time intensive, and so we've focused on these kids who do respond. So I don't have a comparison to kids who don't respond, nor do I have a comparison to kids who have OCD but not pandas. That's a really important question. So these are in our near-term research plan, but I don't have those answers yet.
Thank you, Chris. Thank you.
All right. Good morning, everybody. I'm Ian Odell, a physician scientist in dermatology with expertise in fibrotic skin diseases. And my co-founder is Richard Fulvel, who's a world-renowned immunologist here at Yale, and also co-founder of Alexion. And so today I'll tell you about Plythera, the company we founded last year for using epireglin inhibition to treat systemic sclerosis. So Canaan is our lead investor, and with their help, we have a great management team, including the former CEO of BI, the former global program lead at BI for pulmonary fibrosis, and a pulmonologist who led the OFEV trials for use for IPF. So basically, the Plythera vision is to establish epireglin inhibition for multiple fibrotic conditions. We're going to start with systemic sclerosis, skin fibrosis, as well as SSC-related ILD. But we're very interested in showing translatability to other fibrotic conditions. So systemic sclerosis is considered a rare disease. It affects primarily women in the prime of their life, 30 to 50 years of age. It's rare, but not ultra rare. New medicines are still getting orphan drug designation. And there's two main issues with morbidity and mortality. So hardening of the skin is the major driver of morbidity. And when it affects internal organs, primarily the lung, that's the major cause of mortality in patients. And when that happens, the average survival is about a decade. There's a large market opportunity here, estimated at $3 billion in the US alone. And that's assuming limited price and market penetration and level of polypharmacy. Basically, the women on the upper right are the typically, what they look like when they show up on the upper right, and in the bottom right is when severe disease hits, and that's what it looks like before this patient passed away. So in our translational studies, we did single-cell RNA sequencing of patient skin from my clinic. In our analysis, we identified EGFR-expressing fibroblasts as having all the pathologic hallmarks of driving disease. We looked at, through bioinformatics approaches, we looked at what EGFR ligand is driving disease, and we found that epireglin was the most significantly enriched, not just in our skin data, but also across multiple data sets, including SSC-ILD and IPF lung. Importantly, the cells that express epireglin are a very specific subset of dendritic cells, and those cells, when they're higher in the skin, correlate with worse skin fibrosis severity, and then also higher in the skin is associated with worse lung fibrosis severity. So we really think we're on the right target for improving disease. So we tested whether inhibiting epireglin in skin and lung tissue directly from patients could be as a proof of concept. So in the top row, you can see SSC skin. And this is a patient from my clinic who had previously failed mycophenolate mofetil. And when we treated her skin ex vivo with an epireglin neutralizing antibody, we saw a significant reduction in procollagen 1 N-terminal peptide. That's the left on the graph. But we also saw a reduction in tenascin C and NOV. And those are critical because tenascin C and NOV in other studies in the serum have been tightly linked to severity of fibrosis in the skin. In a patient that died of idiopathic pulmonary fibrosis, we were able to do, since we had more tissue, we were able to do head-to-head trial of EREG inhibition compared to TGF-beta inhibition with an ALK5 inhibitor and nintedanib, which is approved by the FDA. Epireglin inhibition was similar or better than the other two interventions at reducing collagen production. It trended towards a better decrease in MCP1, which is an inflammatory cytokine associated with SSC. And it was actually the only intervention that reduced TIMP1, which is a key regulator of collagen degradation. So this is actually a way in which our intervention could actually reverse fibrosis, not just decrease its advancement. So we went on to develop, with the help of Colton funding, a fully human neutralizing antibody against epireglin, high affinity of 38 picomolar. This antibody has all the characteristics of that can be developable. It inhibits the full-length protein. It's specific to EREG and not other EGFR ligands. It's stable at 4 degrees for more than 6 months, and it has all of the CDR lengths and hydrophobic patches all are in the right place compared to all other FDA-approved molecules. So we're currently narrowing in on our developmental candidate, and we're and that's been taken through microdevelopability at WuXi. So what stands us apart from the competitors is that we're at a completely unique mechanism of action that's rooted in mechanistic understanding of SSC biology. And so we're currently raising Series A funds to take our program through phase 2A trials for proof of mechanism. And then that could lead into a potential follow-up EOCC phase 2B. And that was it.
Yeah, I'm curious to know how this compared to CD19 CAR Ts, because it also works recently.
Right, so there's a recent publication in the New England Journal on using CAR Ts for SSC and other autoimmune conditions. I mean, the clear thing is that there's no control group in that study, and scleroderma is notorious for having spontaneous improvement in certain scores, and so it's hard to tell in that sense. And they used the U-Star AI criteria, which is relatively new, but we're very interested in using that as an outcome measure in our studies as well. Yeah.
Two questions. A huge amount of work, which is exciting. Have you looked for the dendritic cells that you were talking about in other diseases with fibrosis? Because you might actually be able to get proof of biology, proof of concept earlier in a less complicated disease. And I think the reversal piece that you mentioned, you think about ILD, is just given the need. If you could do early markers even of improvement, you might be able to really accelerate your overall program time.
Mm-hmm. Basically every fibrotic disease we've looked at, if it's immune driven in any way, it seems to be there. But yeah, we're.
So you mentioned the potential ability to reverse fibrosis. Have you guys discussed the reduction or elimination of the general immunosuppression?
So the other thing, the critical thing here is that Lilly has an antibody that cross-reacts with our target, not to the same affinity, but they hit therapeutic threshold in their phase 2 studies with no toxicity issues. And so actually we're thinking that this is not going to be as immunosuppressive as the standard of care. And actually it's all looking like it has a very safe profile. Yeah.
Just for the safety profile beyond fibrosis, what are the risks that involve?
Yeah, so this target is mainly expressed by the dendritic cells that are increased in inflammatory conditions and not, it really doesn't have a role under homeostatic conditions. We made knockout mice, they're completely phenotypically normal. The only organ which is expressed is actually in the ovary, because there's this wave of EGFR ligand expression, they're going to ovarian maturation. So if you hit this, there's no problem. If you hit this plus another EGFR ligand, then you get reproductive issues. So we know our antibody is safe and specific, but that's an important facet.
Thank you very much. So our next speaker is Essen Sepik. I'll introduce Essen because she's a new faculty member in the Department of Immunology at Yale. So welcome, Essen. Thank you for coming.
Thank you. Sorry. So my boss, current boss,
Former boss.
former boss, don't say that to Richard, was supposed to give this talk. So I'm rusty on this, but I've seen the slides. So this is our team. You all probably know Richard Thawal, and we have been working on this project in collaboration with Dr. Bill Philbrick and myself. And just to remind you, we will be talking about celiac disease for the next 5 minutes. So what is celiac disease? Celiac disease is the most common autoimmune-like disease. We know the antigen, it's driven by dietary gluten. And in a subpopulation of patients, when they ingest the antigen, it presents with severe intestinal issues. It's a lifetime problem. Gluten avoidance is pretty much the only cure or avoidance we have, but it doesn't work in 1/3 of the patients. So we need therapeutics, and we need these therapeutics fast. And we know it is an immune cell-driven disease, but we do not have a mouse model to study this disease mechanistically, and we don't have a platform where we can test therapeutics. So this is what celiac disease looks like right here. In a healthy intestine, you see these beautiful villus structure, which maximizes nutrient intake, and the immune system ensures that there's tolerance to the continuous challenge of antigens provided by the food we ingest. In celiac blunting, to mechanisms that we don't truly understand, T cells attack these epithelial cells in the line of us, so here, and leads to epithelial cell death and villus atrophy or blunting. We know the antigen, we know the cells that are implicated in this pathology, but we really don't really understand the mechanism of how gluten ingestion leads to this severe intestinal problem. And that's what we are going to try to make. We would like to make a model of human disease using humanized mice, and we would like to use these mice to test therapeutics, either in the context of therapy after or as preventative medicine. So we have multiple problems to solve. The first one we've already solved. In a way, we have created mice that have a human immune system. These mice provide the microenvironment for human immune system to develop, which comprises of both myeloid cells as well as lymphocytes. We know that there is a very strong genetic predisposition in celiac disease. So we have acquired a large set of tissues and cells that we can use to start a human immune system in our mouse models. But we need a couple more parameters. So what we decided to do is go back to the patients and take human celiac disease as a roadmap to guide what we need in this new mouse model. So we need antigen presentation in the right context with the right genes. We need certain cytokines and cytokine signaling pathways. And based on our single-cell RNA sequencing and more recently spatial transcriptomics data, we've identified that IL-15 signaling as well as interferon gamma signaling are essential players in celiac disease. So currently, we're incorporating these into our humanized mouse system to be able to create a celiac disease mouse. So the idea then is once we have this mouse model, when we feed this mouse gluten, we expect to find intestinal inflammation reminiscent of celiac disease. And this will be the first of its kind, a humanized mouse model that models celiac disease in the context of human immune cells. So as I said, I've mentioned that we took the human celiac disease as a roadmap, and it identified different parameters, and I've listed 5 of them here that we're currently incorporating into our mouse model. IL-15 signaling was essential. It is known to activate T cells, but its role is not really known, and it is not known which cell produces or transpresents this cytokine in the context of celiac disease to activate T cells. So we've already made the mouse that has it and incorporated into the Mr. G6 celiac disease mouse that we are generating. Interferon gamma signaling, as well as antigen presentation by MHC class I molecules, is another point of focus. They go hand in hand. We already know that human interferon gamma upregulates MHC class I and the epithelial cells, so we've done that. And we do see this in celiac patients as well. And then finally, we're focusing on 2 additional MHC class II-like molecules or MHC class II molecules. One is being HLA-E, and the other one is the class II that predisposes people to celiac disease. And we've generated mice for both these, but we haven't incorporated them into our final mouse model yet. With that, I'll take questions.
Questions? Were CTLs the primary driver of disease damage? Because that's what you're targeting. You're trying to make sure human CTLs are maintained with the IL-15.
But it's interestingly, the CTLs need to locate into the intraepithelial layer in celiac disease, and that's not really seen in other inflammatory diseases where they can be localized into lamina propria. So what happens in celiac disease is the IELs or intraepithelial lymphocytes that maintain homeostasis are replaced with cytotoxic T cells. We don't understand what drives them to that layer, and we don't really understand what activates them.
If I may, what are there good data on using other known immunomodulators that have been approved clinically in humanized mice to look for efficacy of those immune modulators in a humanized mouse model?
Yes, I mean, I can give an example. My prior research is focused on COVID-19. we've been trying to understand severe COVID-19 mechanisms. One good example would be dexamethasone, which is given to patients, and it's pretty much the only immunomodulator that provided some relief from severity of COVID-19 and reversed symptoms in clinic. And it works as we expect in the humanized mouse model. But what we have been able to uncover in the humanized mouse model was the timing of dexamethasone. It's a broad spectrum immunosuppressant. It suppresses everything from T cells to neutrophils. And if you give it too early, it's detrimental. It kills the mouse. You have to time it in accordance with viral titers. So you have to time it at a time when viral titers go down. And we've been able to uncover that in the humanized mouse model and partially understand. There are other mechanisms, drugs that we've tested, type 1 interferons being one of them, for activating the immune system or suppressing the immune system on the flip side of the things. And they do work in a similar way. Monoclonal antibodies, another example, where maybe not immunosuppression in that sense, but targeting, it's the same monoclonal antibody that you would use in a patient. You can just take it off the shelf, give it to the mouse, because everything in the system is humanized to allow these drugs or antibodies.
Other questions?
Chris, you mentioned preventative medicine versus therapeutic. I don't know if it's too early, but do you feel like one is more promising than the other?
Well, the nice thing is we know the genetic predisposition, right? So it only happens in people which carry the HLA-DQ 2.5 and 8. So preventative medicine is actually possible, and we see it in children as well. So you can start with that in patients with high risk. One family member already having it and having the MHC class 2, that would be a very feasible approach. But we're also interested in therapeutics because it's really difficult to have gluten avoidance, and it doesn't work. So, and there's a huge population of patients already out there that already have celiac disease.
Thank you.
Thank you, Essa. Thank you.
All right. So hi, everyone. My name is Jun Wang. I'm assistant professor at NYU. So today I'm excited to share with you my research in targeting key feedback immunomodulators for immunotherapy of cancer and autoimmune disease. This is my team, including myself as a scientific co-founder. I also have some connections with biotech companies and some startups. I'm also lucky to have Dr. Leo Snumberg to advise my company. He has been a biotech veteran and has been the leader for the immunotherapy field. So what kind of questions we are thinking about? So we try to answer the unmet medical needs for immunotherapy of cancer and autoimmune disease. For cancer, as we know, PD-1 has been successful, but only in 20% or 30% of patients. So in part of the hot tumors, it doesn't really work very well, especially in cold tumors, PD-1 have weak responses. So we have to identify effective targets for PD-1 insensitive tumors. For autoimmune disease, we have lots of approaches to target B cells and also many targeted approaches for cytokines. So we don't have effective therapies for T cells and myeloid cells. So altogether, this is a big field. We have over $10 billion market value each year. So our solutions, so we think PD-1, PD-L1 is not just like immune checkpoints. It's not just like immunosuppressive molecules. More importantly, it represents a negative feedback loop. What I mean is tissues, for example, tumor cells, since T cell coming, cytokines, abrogate PD-L1 and suppress T responses. This will make sure this targeting will be restricted to disease microenvironment. Then we can use blockade to enhance T responses. For cancer, we can use agonists to shut down T cell for autoimmune disease. So we have been thinking about what kind of other targets, PD-like targets, reshaping can be important for the microenvironment, forming negative feedback loops. Then we can repair the immunity in the disease lesion. We can use this to target cancer and autoimmune disease. We're thinking about two questions. One is PD-1 actually mostly targeting exhausted T cells, same subsets. What kind of targets in other T cells, especially memory T cells? And the second question is type interferon is very important for our immunity, either autoimmunity and tumor immunity. And we already have drug. Recombinant type interferon has been proved for cancers, has been proved for multiple sclerosis, but it's very toxic and modest efficacies. Can we identify some kind of checkpoint modulators, feedback modulators in the disease lesion, shut down type interferon, and then we can use that as a target to recover type interferon? We already identified a molecule called myeloid inhibitor of type interferon. If we lock it out in macrophages, you see enhanced type interferon, very dramatic difference. And then this is a molecule induced by STING agonist. So that means STING agonist trigger type interferon, at the same time trigger these suppressive molecules to shut down type interferon. It's a selective feedback loop. This molecule expressed on lots of tumor cells and especially melanoma and some of the cold tumors, high expression associated with worse survival. It's a good target. And then we already have antibodies. Itself has single agent activities. This effect is through type interferon because interferon blockade bring back the difference. And these antibodies combined with PD-1 works really well. As you see here, all mice survived. And then in cold tumors, it works much better, even in the presence of PD-1 LAG-3 compilation. So it's a good target for cancer. At the same time, because interferon is therapeutic for multiple sclerosis and the Burkitt's disease, so we already have some preliminary data showing antibody blockade or knockout mice can really control the multiple sclerosis mouse models. So this is kind of interesting for autoimmune disease as well. The second program is we identify molecules called PDME, PD-1 mutual exclusive genes. This molecule is very different from PD-1 LAG-3. As you see here, this is from melanoma single cell data. PD-1 LAG-3 just expressed on exhausted T cells, and most of the other subset memory T cells are active of PD-1 LAG-3. We found this molecule is very different. We also have blocking antibodies showing single agent activities, and then in combination with PD-1, it works really well. We believe this is a real synergy because we're targeting both exhausted T cells and other T cell subsets, for example, like memory T cells. At the same time, we are generating agonists for autoimmune disease. To summarize, we are targeting something beyond immune checkpoints, immune feedback modulators, selective feedbacks. And then we will have proof concept with our antibodies cross-reactive to both human and mouse. So we're thinking about humanize, move to INDs. And then this has exciting commercialization opportunities for PD-1 resistant cancers and also autoimmune disease focused on myeloid cells and T cells. Thank you so much. Looking forward to comments.
I was wondering whether you've had the chance to look at tissues from patients either who've been unresponsive or have responded and then relapsed, and what dynamics you observed.
Yes, we did find, especially the interferon inhibitors. So we do find some PD-1 resistant cancers have more upregulation because this molecule induced by inflammation, STING agonist, or toll receptor agonist. So that's why we really believe you will form some kind of feedback response upregulated in the disease resistance or inflammation.
Yeah. Have you identified a potential ligand for MIFN?
That's a very good point. Actually, we have some data right here. It could have two mechanisms. One is it might have cis or trans, like it binds to itself, like a dimer. That's one. Second is they do have the endogenous ligands. We already know which epitopes it can potentially bind to. And we already know the antibody epitopes right now.
I'd be curious to understand how you're designing agonists for some of these pathways.
That's a very good point. So I have a question. Yeah, so I do have some experience working with checkpoint agonists. Right now, most of the checkpoint agonists, you need the cross-linking. That's something we're working on.
So there are other therapeutics for interferon-responsive diseases, multiple sclerosis being a classic example, hepatitis C being another example where there is other therapeutics. Behçet's, however, there's no therapy for Behçet's, as best I understand, as a rheumatologist. So are there really strong data that Behçet's is an interferon-responsive illness?
Yes.
No therapy. This is a very common illness, of course, across the world, not so much in the States, but in other parts of the world.
Yes, I think it can be very exciting to look at.
Are there animal models of Behçet's?
Yeah, I do have some, but the question here is how close it can be to the human disease, but there are some models there.
It's a huge therapeutic unmet need, this illness, and it's very disabling illness as well.
Yeah, so I'm getting excited for this target.
Yes.
All right. Thank you.
My name is Dr. Jonathan Miner. I am a rheumatologist and scientist at the University of Pennsylvania Perelman School of Medicine. And today I'm going to talk to you about therapeutic targeting of the TREX1 cGAS STING pathway. Our team includes me, a medicinal chemist named Noreen Niamati at the University of Michigan, Sean Clark, who is CEO of our company that was recently started called Frameshift Therapeutics, and his father, Bob Clark, a businessman, founder of a company called Clayco. Bob's wife and Sean's mother died from a rare genetic disease called RVCL that is a consequence of mutations in a gene called TREX1. RVCL is a very rare disease, only a few hundred families, but the majority of the patients have just one mutation. It mimics lupus. The disease is universally fatal, and there is no treatment. The patients get sick around age 40, and then all of them die within 5 or 10 years. My laboratory recently discovered the mechanism of the disease. Our therapies that are in development, we believe, are likely to also be useful for common diseases, including cancer and autoimmunity. And finally, gene therapies and effective TREX1 inhibitors were recently developed by us. So an overview of the work we do in my laboratory is we make mice with human genes and human disease-causing mutations. We use them to study mechanisms, but also to develop and test various therapeutic strategies, including small molecular inhibitors and gene therapies delivered as mRNA lipid nanoparticles or AAV-based therapies. This disease is caused by mutations in TREX1, which is a DNase that's localized in the cytosol. It's very important in regulating the STING pathway, which we just heard about. Activation of the STING pathway induces interferon responses and T cell and NK cell mediated immunity. Inhibition of TREX1 activates the STING pathway. So, as was alluded to earlier, STING activation is currently being pursued as a form of tumor immunotherapy. So STING agonists can cause tumor aggression, and this is achieved by activating T cell and NK cell responses. TREX1 inhibition has a similar effect. Just to give a highlight of some of our approaches, we have screened a DNA-encoded library. So this is a library of small molecules, each one of which is linked to a unique DNA barcode. We screen these by pulling them out based on binding to TREX1. And out of 7 billion molecules screened in experiments, we pulled out around 1,000 of them, and many of them are currently being produced at WuXi. But we also have other inhibitors that we've already tested using our mouse models. So in this case, we have a mouse model where we can turn on the TREX1 gene, and we can see the effects of TREX1 in primary cells from the mice or within the animals. And we can confirm that some of our TREX1 inhibitors block toxicity mediated by, in this case, an RVCL-causing TREX1 mutant. These inhibitors also work against the full-length normal TREX1 protein. Our gene therapy utilizes prime editing, and using prime editing, we can convert a mutant protein into a normal protein, and we have developed and tested numerous delivery methods for this gene therapy. That work is ongoing, and we have a provisional patent for it. So our gene therapy is confirmed to be effective in vitro. We're working on testing in animals right now. We have a co-crystal structure of one of our inhibitors with the TREX1 protein, which allows us to see how inhibitors interact with the protein and improve them further. POP inhibitors are being evaluated for pharmacokinetics. Our goal is a clinical trial of the gene therapy and hopefully the inhibitors in the next few years. So we have numerous competitive advantages based on the tools that we have, including the co-crystal structure, unique animal models expressing the human TREX1 gene, our DNA encoded library screen, TREX1 targeted gene therapy, and also unique access to a TREX1 associated disease population with highly committed support from donors at the Clayco Foundation. However, we've not yet determined which compound will be our lead, but because of the tools we have, we're able to rapidly screen and expect to determine that soon.
Please, Colton.
I'm just curious about the cell type expression of TREX1 and where you need to get the gene therapy, and if you've been able to demonstrate that.
Great question. So TREX1 is ubiquitously expressed. The affected cell type in this rare disease is the vascular endothelium, which provides some interesting challenges. But we are collaborating very closely with Vlad Muzikantsev at the University of Pennsylvania, who has technology, and he's also supported by Clayco, technology for efficient delivery of lipid nanoparticles to the blood vessel wall. Also working closely with Drew Weissman on this, and so we're optimistic that we're going to be able to deliver to the vessel wall, although it's possible that additional innovations will be necessary.
Ask one more question. How much of the editing, like in other words, how much non-mutant protein do you need, wild type protein do you need to overcome the expression of the mutant? So in some diseases you need, 100% of the.
That's a great question. So the key concept here is the mutant protein is toxic above a certain threshold. So lowering TREX1 expression below that threshold, we predict would be protective. What we don't know yet is what percent of cells must have. In what percent of cells must we correct the mutation in order to prevent disease? That's an open question and a very important one. Yeah. Mm-hmm.
If gene therapy is ultimately cure for the disease, but if overexpression of the type 1 interferon is really pathogenic, then can you use the anti-interferon receptor antibodies for this disease?
Very interesting question. Remarkably, in this disease, what's happening is that there's a toxic TREX1 protein that relocalizes to the nucleus and starts damaging the genome. So part of why this rare TREX1-mediated disease is untreatable is it doesn't respond to immunosuppressive therapies at all. But it does provide a platform for rapidly screening TREX1 inhibitors, and the inhibitors not only prevent the toxicity of the mutant protein, but would likely be useful in other diseases. Mm-hmm.
So is this target? This is the dominant autosomal?
It is a dominant negative. It is not a dominant negative. It is a dominant toxic gain of function that mislocalizes to the nucleus.
I see. So PROTAC inhibitors would be a potential therapeutic.
Yeah, our DNA encoded library screen for binders is actually we're using that as the platform for PROTACs. Yeah. Mm-hmm.
I may screw this up, so the ophthalmologists in this audience, please correct me, but I think there's a disease called peripheral vitreoretinopathy, which PREX has been indicated as a driver of that disease. Are you aware of that?
I am not aware of the link between those two diseases, but that's a very interesting comment, and I'll definitely look into that. Yeah.
I think you're correct, Gene. It's also familial chilblains. It was discovered by a guy named John Atkinson at the Washington University in St. Louis, but it is a TREX1.
Chilblains, so the Chilblains mutants are dominant negative, totally different from what we have here. Yeah.
Thank you. Yeah. Okay. Thank you very much. Yeah, we can do 5 Zoom presentations. These are we'll get going, but I think we'll have the speaker live so we can ask the speaker questions.
Good morning. My name is Christoph Ellebrecht. I'm an assistant professor of dermatology at the University of Pennsylvania. I'm very excited to be here today to show our data and to share our opportunity with you on innovative therapies for chronic skin diseases, targeting skin-homing T cells for long-lasting remission. As mentioned, I'm faculty at the University of Pennsylvania. I'm a scientific co-founder of Cabaletta Bio. I am not working for Cabaletta Bio, and what I'm presenting today it does not have anything to do with Cabaletta Bio. Nevertheless, I'm an inventor on patents licensed to Cabaletta, Novartis, and Community. And I'm a physician scientist. I'm a dermatologist by training, running a research lab with experience in advancing novel technologies from discovery to IND. If you want to contact me, this is my email address or my lab website. I'd like to start by saying that the skin is the most commonly affected organ in autoimmune and inflammatory conditions. The most common autoinflammatory conditions are listed here on the left, and they affect millions of Americans with a sizable market size listed here. So this is a great opportunity if we can improve treatment of these conditions. While they look clinically distinct, as shown here on the right, they all have macroscopically one feature in common. If we look at healthy skin, we don't see any inflammatory infiltrate. However, in skin and autoimmune or in autoinflammatory conditions, we see these little blue dots, and these are T lymphocytes. All of these conditions have in common that they are driven by T lymphocytes that enter the skin and cause tissue damage. And once they are in the skin, they are really stubborn, they are really difficult to eliminate, and current standard of care therapies cannot eliminate these cells. That's why one of the limitations of standard of care is the inability to eradicate pathogenic T cells and the risk of relapse upon treatment cessation. And this was shown in a study published last year that after IL-23 blockade, shown here in blue, and this is standard of care in psoriasis treatment, for example, we still are able to detect T cells in the skin and other T cell subsets, and you need a greater than 90% reduction, shown here, greater than 90% reduction in IL-17 producing cells in order to count as a strong responder. Only a 75% reduction will not suffice. You will count as a poor responder. So these cells are really potent at causing tissue damage and also are really difficult to eliminate. That's why we want to develop new therapeutic approaches. We want to target skin-homing T cells specifically for sustainable remission and chronic inflammatory skin diseases. We developed a new screening model, which is a completely humanized model using human skin grafts on immunodeficient mice. Then we inject T cells, and over time, we see these inflammatory infiltrates in the skin, shown here, but also in the spleen. So the T cells have to make a decision whether they go into the skin or stay circulating throughout the mouse. So that gives us the opportunity to screen what are the best therapeutic targets to specifically deplete T cells in human skin. And we have done one of these screens, and on the x-axis, you see basically the ranked therapeutic targets, and on the y-axis, you see the x-fold depletion of T cells from inflamed skin. And this is the current standard of care, 10x, 90% reduction. And we were able, by targeting novel therapeutic targets with different modalities, to deplete T cells from inflamed skin up to 800-fold. And we have a lot of new therapeutic targets that haven't been targeted before, and that's why we are convinced these are promising avenues for furthering inflammatory skin disease treatment. That's why we patented most of these targets listed here. If you want to find out what these targets are, please feel free to reach out. We are happy to share these targets with you and see how we can move them forward. The idea is that when we compare the landscape of systemic therapies for inflammatory skin diseases, we already have very potent therapies. Cytokine neutralization, cytokine signaling modulation, for example, JAK inhibitors, quite potent. The problem is, once you stop these medications or lower the dose, the persistence is just relatively poor, the persistence of the effect. So we hope that by targeting new therapeutic targets that we patented, we not only can increase the potency, as we have shown, higher fold depletion of T cells from inflamed skin, but we can also increase the persistence of effect so that we may be able to actually have a curative treatment in the future. If you'd like to learn more about this, please feel free to reach out. My contact again, we are happy to share these targets with you and the modalities, obviously, and I'm happy to take questions. Thank you for your attention.
So questions for Dr. Elbrecht? I'll probably repeat that. I'm not sure you can hear it. Please.
Question: Are these small molecule targets or antibody targets or mixture of both?
So, Chris, can you hear me okay?
Yes, I can.
Yeah. So the question is, are these small molecule inhibitors or antibody targets? What's the strategy here?
We have both. So some of these we can target with antibodies. Some of them would require small molecules or other modalities. We're platform agnostic here.
Other questions? Yes.
Do you say something more in terms of the specificity of the targets that you've identified? And are they targets that are really more unique to the T cells that are in the skin? Or do you anticipate a systemic effect? And which is the more important?
So, Chris, can you say more about the targets you're looking at and the specificity for skin homing T cells as opposed to other T lymphocytes?
Absolutely. We believe, so there are two options. Either you compartmentalize the activity of your treatment, or you find something that is unique to your population in a lesion compared to circulating and non-lesional T cells. And we discovered both.
Okay. Another question? Yes.
Are there any positive controls like integrin inhibitors to validate the screen?
Absolutely. So we have positive controls in terms of T cell signaling, cytokine signaling, and migration chemotaxis inhibitors. Yes, like sanity checks, basically, whether the screen is working.
Yes.
Thanks for the presentation. Great. Insight has a topical phosphoinositol kinase 3 delta inhibitor. Have you tried it?
We have not tried it. It's quite interesting. If you, yeah, it is quite interesting. It might be something to pursue, but we haven't tried it in this model. Yes, it is interesting. If you overactivated T cells in a genetic syndrome, are more likely to be in central compartments as opposed to the lungs or the skin. So it's interesting how you modulate this. PI3 kinase delta subunit. So it's certainly an interesting one to look at.
One final question. So T cells in the skin that home to the skin also have homeostatic function in terms of barrier protection and so forth. Are there worries about off-target effects here in terms of disruption of normal homeostatic function of T cells that home to the skin, absent pathologic indications?
Yes, that's why it's, in our opinion, important to distinguish what is lesional and what is non-lesional skin. And then do you find a target that is unique to lesional skin and also unique to circulating or compared to circulating T cells?
Okay, thank you, Chris. That was good. Thank you.
Hello, everybody. My name is Rahul Kohli from the University of Pennsylvania, and I'm pleased to be talking to you about the next page of CRISPR genome engineering. I'm speaking to you on behalf of my team of collaborators and co-founders of NextPage Incorporated, including Junwei Shi, Shelley Berger, and John Wary, who are respectively experts in CRISPR, epigenetics, and T-cell biology. I'm a biochemist with an interest in DNA-modifying enzymes and processes. Our focus as a team has been on thinking about the challenges posed by current CRISPR delivery strategies. We know that there is great potential in cellular therapeutics, particularly if they can be further engineered at genetic targets. However, the current methods show limitations. The way that CRISPR genome editing machinery is introduced into cells of interest is typically through viral vectors or through physical electroporation-based or lipid nanoparticle-based approaches. Each of these methods suffers from certain limitations, including the fact that viruses can insert randomly into the genome. Physical methods can cause toxicities by the act of electroporation or variability in the cell types. We decided to reimagine this challenge by thinking about the fact that the proteins themselves
can be directly delivered into cells through engineering strategies. And our cell-penetrating approach is assisted by peptides that allow for this delivery into cells, out of endosomes, and into the nuclei. This peptide-assisted genome editing machinery, or PAGE, forms the basis of our foundational technology and patents. In published work, we've demonstrated that in critical cell types like CAR T cells, as short as a 30-minute incubation of our proteins with cells allows for direct cellular uptake and remarkable genome editing efficiencies of up to 98%. To give you a sense of the head-to-head strengths of this methodology against commonly used methods like electroporation, we find that PAGE is non-toxic, that allows for a higher recovery of the cells that have been edited, and most strikingly, that allows for minimal perturbation of those cells. Shown at bottom right is the idea that electroporation results in thousands of genes that are up or down regulated after shocking the cells, while proteins being delivered directly through cell-penetrating strategies results in no perturbations to the transcriptome. The opportunities opened up by non-toxic, high recovery, and minimal perturbation are that we can now edit fragile cells. A model for this comes from exhausted CD8 T cells that can be generated in the LCMV model. And in this setting, in the presence of our Cas machinery, as well as our assist peptides that make up the constituents of our PAGE technology, we can now edit these exhausted T cells for the first time. This was previously unachievable in ours and others' hands through the conventional approaches like lipid nanoparticles, electroporation, or retroviral-based approaches. These cellular therapeutics are opening up new opportunities for commercialization and for expansion of the technology. We've identified tumor-infiltrating lymphocytes as our primary target for our first product. And to move towards this end, we want to demonstrate that such fragile cells can be cultured and manipulated without toxicity. The protein-based technology opens up new opportunities for more complex genome editing beyond simple nuclease activity, allowing for engineering in ways that can allow for these cellular therapeutics to take on their true potential. And the fact that our technology only requires cells and protein means that strategies such as direct pheresis from a patient, engineering, and reinfusion are now all opened up as next avenues for pursuit inside of NextPage Therapeutics. We've identified our goals for our $10 million seed round to be the production of our system, the transition of the system into new editing technologies, and a specific focus on positioning ourselves for that first product in tumor-infiltrating lymphocytes. I appreciate this opportunity to share with you more about NextPage Therapeutics. My contact information is below, and I'm happy to take questions or entertain other interest. Thank you.
Questions? Please, John. You showed the reduced perturbations of DNA in some cells. Do you have reason to think that this method of transfection of the CRISPR will have reduced off-target gene editing?
So, Rahul, I don't know if you heard the question, but the issue is you showed there's less perturbation of the genome with your new technology. Do you anticipate there'll be fewer off-target effects using this approach as opposed to standard CRISPR approaches?
Yeah, absolutely. So one reason for off-target activity is the continued expression of genome editing machinery. With protein delivery, we have the end product, the editor itself being introduced into the cells, and it itself is going to have its lifetime is what's going to be what determines off-target activity, as opposed to delivering nucleic acids that are going to make more protein that is potentially able to have more off-target activity downstream. Though that wasn't a specific focus, we do know that the lifetime of our editing machinery is short, and we anticipate that will decrease off-target activity as well.
Other questions? Yes.
Could you say something about the nature of the protein and whether changes to that protein that you're using actually could affect specificity in terms of reaching different cell types, or is it a more general process?
So, Rahul, could you say more about the protein
you're thinking about using, and would changes in that protein, for example, affect specificity of the target?
So the published data is on Cas9, Cas12a engineering, both as proteins and as ribonuclear proteins. The protein engineering has taken place outside of the scaffold of the core enzymes. These are added domains that are influencing the uptake, in addition to the peptides that are added in trans to allow for uptake and escape from endosomes. So the anticipation, we do not anticipate any effects on the core catalytic functions of the enzymes.
Sorry, just in terms of whether though do different types of protein variations affect the type of cell that you might in terms of specificity? Sorry, I don't think I.
Yeah, so cell type specificity, I think I heard that extension of the question. So in the demonstrated, again, published data, we've demonstrated targeting multiple cell lines, hematopoietic cell lines, as well as stem cell progenitors. We do anticipate that some of the idea about what it takes to get for a cell-penetrating protein to be taken up by a particular cell, what peptides will assist, may differ from cell to cell. The conditions may differ. And one of the objectives in next page is to understand the full breadth of primary cells that one might want to edit. We've targeted the most, I think, high-value types of cells where therapeutics are already being developed, but understanding the full scope of specificity and necessity for any modifications to the existing protocol will be something to consider.
Other questions? Okay. Yes, please.
Yeah, not a science question. So, what's the business approach here? Are you intend to sell this as a kit to be used by other research, or what is the business approach?
Yeah, Rahul, I'm not sure you heard that. We're going to repeat the question if you give us a second. Thank you.
Yeah, so I'm wondering what is the business strategy here? Are you going to sell this as a kit for other researchers to be used, or that's my question?
Great. Yes. So the business strategy here is that for reagent use, we intend to scale up production of the protein and to share. A goal of the science is to disseminate it, and reagent sharing will be an early objective. But at the same time, we will maintain rights over therapeutic applications, which will allow us to either pursue partnerships. We've demonstrated superiority to methods such as electroporation that are already being used in clinical trials, as well as pursuit of our own therapeutic targets. So it's a multi-tiered strategy and one that's still being formulated and open to input from the business strategy side.
Okay. Final questions? Rahul, thank you very much. That was great. Thank you.
Thank you.
So we're going to change time zones, so bear with us. The time zone shock, we're going to go to Tel Aviv.
Hi, I appreciate the opportunity to present here today. Eva Therapeutics is a spin-off company from Tel Aviv University. We aim to target the sweet spots of solid tumors with breakthrough immunotherapies. Basically, glycans are unique targets that make up ideal targets for cancer therapy. My name is Vered Pader-Karavani. I'm a professor at Tel Aviv University with over 20 years of experience in glycobiology, antibody engineering, and cancer immunotherapy. My co-founder and CEO is Ya'ara Tziporen Ochayon, which has 14 years of experience in cell and gene therapy, particularly in manufacturing GMP-grade viral vectors and clinical GMP for cell therapy products. Cancer is a devastating disease affecting over 20 million patients worldwide. CAR-T immunotherapy is a unique approach that takes the patient's blood, engineer the T cells outside of the body to arm them with unique entities that would allow them to bind to the cancer cells specifically, and then they get stimulated to kill those target cancer cells. While this approach is successful, it currently only works for blood cancers in the clinic. which make up only a small fraction of the cancer burden, while the majority, over 90% of the cancer burden, is of solid tumors that don't have druggable targets. We propose to target sugars. Apparently, every living cell has this thick layer of sugar coating that is critical for their cell survival. Cancer cells have very unique cell glycosylation that make up tumor-specific glycoantigens that are ubiquitously and homogeneously expressed on the cancer cells. Our GlycoFind proprietary technology allows us to identify such neoglycan targets, to engineer glycobodies against them, and to screen them in vitro with our unique know-how and approaches. Once we validate our candidate products in vitro, we move to validation in vivo, and then that allows us to move for application-tailored, optimal, specific binders. Currently, we have 2 candidate products that are protected by patents, and we hope to take them for next steps to the clinic. Just to give you an example of how we work on that, here we have a glycan microarray screening of our lead 201 glycobody, and we show that against 88 different glycans that can be expressed on normal and cancer cells, our glycobody specifically binds to our unique cancer glycans and its derivatives, but not to the other glycans. When we use that glycobody to examine the expression of the target in cancer patients, we see homogeneous and ubiquitous expression as through this brown staining, but not only in the tumor, but not in the normal nearby tissue. And we see that in a range of carcinomas, in a wide range of different carcinomas. We then take our lead candidate products into engineering the CAR-T, and compared to the control that was not armed with the CAR-T construct, we see that only the CAR-T product has a very efficient killing of the cancer cells and efficient survival, but not to the control adverse-used T cells with no survival. This basically provides proof of concept of our technology and approach. In terms of competition, while there are many clinical trials currently running that use CAR-Ts against solid tumors, there are only few glycan targets that have been explored, and our targets are different than those. Other companies use different targets than ours, or use different modalities, or different approaches. In terms of our 3-year plan, in the first year, we aim to transfer the technology from academia to the clinical setting, to then submit the regulatory package first in Israel, but then later to the EMA and FDA, and hope to start the first in human clinical trial first in Israel by year 3. Naturally, our second candidate product will follow a similar schedule. To summarize, our breakthrough technology is targeting the untapped neoglycan field of sugar coating of cancer cells that addresses a huge unmet need for solid tumors. If you want to hear more, feel free to contact me or Yara in the following emails, and I appreciate you for listening. Thank you.
Thank you. Questions? Yes.
So you mentioned that the glycan is relatively homogeneous between. Thank you. Definitely need this. So you mentioned that the glycan is homogeneous, between patients and within the tumor. Could you elaborate on how you're looking to identify the patient population?
Great question. So actually, our GlycoFind technology allowed us to generate antibodies that we could use to engineer CAR-T, but we could also use these antibodies in biopsies of patients, which allows us to better select the patients and fit the target to the CAR, or the CAR to the target.
Do you know if these glycans are necessary for cell function and if they're stable? And what's behind my question is, are the tumors going to be able to rapidly evolve away from the targeting antibodies?
So actually, the sugar coating of the cancer cells is a way for the cancer cells to evade the immune system in a way that they cover themselves with much higher levels of the sugars compared to normal cells. And in general, both for normal cells and for cancer cells, this sugar coating is critical for their survival. If they lose these glycan targets, they will be dying, and of course, this is another way to approach cancer therapy by removing cell surface glycans, as has been done by other companies. Actually, just one company that does that. But basically, we target the protection mechanism of the cancers. So no, they will not lose that overgrowth.
I'm just curious why you chose to go with the CAR T approach and not use your glycofine technology on something like a T cell engager format, or if that's something you're thinking about for the future.
Yes, actually, the antibodies that we have generated can be used for any application that you can think of with a targeting moiety. So we can use ADC, we can use bispecific antibodies, T cell engagers, CAR T. The reason we thought CAR T would be the best modality to begin with is that we think we have advantage with a synapse formation that happens with the T cells when it comes to binding to a very wide and high expression of the targets that also avoids non-specific binding to other areas that could happen, for example, with an antibody, or the signal will rely on the avidity of the signal with the CAR T.
Does the presence of the sugar coating on tumors affect the tumor microenvironment that might inhibit CAR T proximity to the tumor cells?
The cancer cells actually use this sugar coating to generate the inhibitory microenvironment. So we are targeting that stemness of the negative microenvironment. Actually, all our immune cells have receptors that bind to these sugar coatings and then send a negative signals. Most of these receptors have I-teams. So when we target the glycans, we also block the inhibitory engagers.
Other questions? Thank you, Vera. Thank you, Yara. Thank you very much. So we have two more virtual presentations before we conclude this session.
I'm going to talk about small RNAs as early predictors for preeclampsia. My name is Noam Shomron. I'm a professor at Tel Aviv University, head of the Functional Genomics and Digital Medicine Innovation Lab. I lead a team of about 30 scientists. I have established several biotech companies, raised money for them, have publications and patents related to our work. I have a wonderful team with me, Professor Eran Hadar, an OBGYN, Director of Maternal Fetal Medicine at Helen Schneider Hospital at Rabin Medical Center, and Dr. Liron Yoffe, a Senior Postdoctoral Research Associate at Weill Cornell Medicine in New York. The birth of a baby, how exciting, the happiest moment in your life. There are close to 4 million happy moments like this just in the USA. Unfortunately, not all end up as happy as this. About 200,000 births will end up in preeclampsia. Preeclampsia is one of the leading causes of maternal morbidity and mortality in the world. Preeclampsia is hypertension that generally occurs after week 20 of gestation. It can cause chronic disability and even death of the mother and the babies. Globally, about 12% of mothers die only from preeclampsia. Some are immunological related, others are linked to increased risk of cardiovascular diseases and type 2 diabetes in the mothers later on in life. What if I told you that we have a crystal ball that can detect preeclampsia ahead of time? And what if I told you that we have a magic wand that can prevent it? Well, we actually do. The current standard care is either risk-based approach supported by maternal demographic history, which is commonly used in the US and Israel. It is simple, cheap, but not accurate. The other method is a multi-marker algorithm, which is common in Europe. It's based on Doppler, biochemistry, and complex because it is time-consuming, 2 blood tests and Doppler in the clinic. As you can see in the table below, the sensitivity, the true positive rate, is not high, meaning it misses real cases of preeclampsia. In our lab, we work on small non-coding RNAs. These RNAs float in our cells, and they regulate gene expression and pathways. They are informative, indicative of epigenetic changes. They were shown to be involved in many diseases. They can be used as markers, biomarkers, and our expertise in the lab is identifying them. We have shown in close to 100 scientific publications that we know how to profile small RNAs and use them as biomarkers or insights into disease etiology. We ran a pilot experiment where we followed hundreds of women where the first blood test is taken at week 10 and stored in the freezers. At the end of the pregnancy, we recorded those that suffered from preeclampsia, went back to the freezer, took the samples out, profiled RNAs, and compared those to controls. We found 25 small non-coding RNAs as molecules which could indicate future preeclampsia much before there's any other signs. Our sensitivity and specificity is 0.7 and 0.8 respectively, but more importantly, we have shown the power of small non-coding RNAs in predicting preeclampsia. Once we add additional features onto that, we will reach a much higher performance. This is our crystal ball. Our magic wand is prescription of low-dose aspirin, if given daily before week 16, will prevent most cases of severe early preeclampsia. We have published our work and we have filed a patent which we received recently. In terms of commercialization, there are millions of annual births worldwide. Just in the US, $2 billion are spent on these babies and mothers in the first year following delivery. We have a few options for future products: a screening test for all on-site in the clinic in a hospital, a service in a central lab, or a standalone algorithm. You send us the data, we send you the predictive value of RNAs in detecting preeclampsia. It could be diagnostic, it could be screening for all pregnant women. Preeclampsia is currently tested through a combination of clinical evaluation and lab tests. Blood pressure measurements, urine tests, blood tests to assess liver function, kidney, and platelets, fetal and mother monitoring with Doppler in order to make sure that the baby and the mother are developing well. In terms of molecular, one company offers preterm birth detection using mRNAs, not small non-coding RNAs, though the signal comes mostly from the second trimester, which is at a later stage of pregnancy and has a limited performance. Our plan is to collect samples, and in order to increase our performance, we will also add electronic medical records onto this data. We have started collecting these samples in a hospital in Israel, and we are seeking additional partners. In summary, preeclampsia is devastating, life-threatening, and costly. Our crystal ball is based on small non-coding RNAs in the blood at week 10. Our magic wand is aspirin. We believe that we are the right team to drive this project forward. Thank you.
Questions, please? No, hi, this is Joe Kraft. So when you draw the blood for the diagnostic test, you're getting immune cells, you're also getting circulating endothelial cells. So which cell type expresses the small non-coding RNAs, and is it specific to endothelial cells, for example? And can you give us a few more clues about what those are?
We are actually looking at floating RNAs in the plasma and in the serum. We would like to add later on additional types of cells which we can extract the RNA from and add them to our predictors. The first thing we wanted to do was extremely naive, just taking the RNAs in the plasma.
Yeah, because arguably, I don't know this field, but arguably the abnormality in preeclampsia might be expressed in endothelial cells, given what the clinical phenotype is. Therefore, if you could select for those, would your specificity and sensitivity of your assay improve?
Well, we do hope it will. Furthermore, preeclampsia is probably not one disease. It's more than one disease. Depends on at what stage it developed and the outcome as well. Even when we look at RNAs, we could see that there are different types of RNAs. In some cases, we see a majority of RNAs are related to angiogenesis. Some of them are related to hypoxia. Others are related to changes in tRNA or mitochondrial RNA. So we believe that we might be able to separate it to more than one disease or more than one types.
Question, please.
Yeah, so I read about the baby aspirin discovery, which has been known for quite a while. What are the risks of prescribing baby aspirin for all pregnant women?
Did you hear that okay, Noam? What's the risk of just prescribing aspirin for everyone?
Okay, so even, I can't remember the exact numbers, but here I speak obviously with many OBGYNs and physicians here. They tell me that giving someone that you don't have any indication for a risk, which is mostly in the first pregnancies, wouldn't work well with pregnant women. You can't just give those aspirins to most women if you don't know what the risk. In particular, in first-time births, where many of these cases come with no risk at all. With the second one, obviously, it's easier, but none of the physicians would prescribe it if you don't have some indications or some molecular markers that would say, okay, there is a risk here. We would like this benefit of preventing preeclampsia with a somewhat risk of taking aspirin.
Yes, yes. A question over here, yes. Yes. Oh, yeah, please.
I want to follow up on Joe's question because this is a good one. If you looked at endothelium and you looked at correlative changes in mRNA expression, do you see some? What do you see? That's my question.
Okay.
Do you see?
I agree. These are very good questions. We are partitioning now the different types of cells in the blood and looking at the mRNA inside. The reason why we went for microRNAs at the beginning and the plasma, because it was accessible. We do have a platform for this analysis. We believe that going up the chain of regulation of genes and looking at microRNAs sometimes could be better. They could be better markers rather than looking at the mRNAs. Looking at the mRNAs could give us maybe more indication about mechanisms, but if we look at the microRNAs, we could think about how these regulate many types of cells, many types of genes in cells downstream from them, and that would certainly would be our next step.
Thank you for a really exciting presentation. We need more work in women's health and for pregnant women. The possibility of a screen for detection, early detection, is really exciting. I would be cautious about the treatment with aspirin, though, without a lot more research into that and your recommendations, both for treatment early in pregnancy and after 20 weeks. There's a lot more thought to be put into that. Thank you.
Yes, I agree. I want to mention that we are developing a marker for GDM, gestational diabetes mellitus. And, I came to the physicians and I said, why don't you just tell everybody to eat healthy food, and balanced food? And I said, no, you have to bring a marker to indicate you are at risk in order to change lifestyle.
So one more question.
Does your microRNA sequencing from serum get exosomes? Does it get RNAs inside tissue-derived exosomes?
Okay. Or extracellular vesicles or exosomes? So,
extracellular vesicles.
So we tried it with and without extracellular vesicles. We see that we also get a nice signal without isolating the extracellular vesicles, which might make the process easier for isolation and downstream analysis.
Final question.
Sure. Just an observation, given the concerns raised about giving universal aspirin treatment in this case, I'm wondering if you're aware of some Massachusetts-based biopharmaceutical companies working on novel treatments for preeclampsia using RNA interference, because I wonder if your detection method might be a useful thing to use together with it, including for clinical trials. The company Comanche Biopharma is developing an siRNA against a protein called SFLT1, which is elevated in the bloodstream of women with preeclampsia coming from the placenta.
Okay, I wrote that down. I'm not aware of them. I know that anything related to pregnancy and treatment, you should be extremely, extremely cautious, and therefore using a drug such as aspirin would be, at least conceptually, easier probably to deliver than this treatment, obviously, unless it's extremely life-threatening.
Okay. Thank you, Noam. That was great. Thank you. So our final speaker is Uri Nevo. He's the director of the Colton Center in Tel Aviv. So Uri.
Thank you, Joe. Hello, everybody. Yes, my name is Uri. I'm a professor here in Tel Aviv University in the Department of Biomedical Engineering. I also, as Joe said, served as the chairman of the Colton Center for Autoimmunity here in Tel Aviv University. And this will not be an immunological work. I will present to you how an interest in immunology and in chronic endocrinological diseases can lead to interesting inventions in engineering. The bone marrow is a critical tissue in many chronic diseases, but typically it is not easy to access the bone marrow for monitoring or for screening diseases. Nevertheless, MRI research has shown the value of probing the bone marrow in different applications. For example, the work presented here of the team of Martel et al. Demonstrated their ability to find differences in the composition of the bone marrow, being more fatty in patients with lupus compared to healthy controls. My team demonstrated the ability to monitoring fatty bone marrow in an animal model of osteoporosis by ovariectomy in rats. Furthermore, we demonstrated that although measurement of bone marrow is known to be significant in spinal vertebrae, these changes can be detected even in long bones as the radius bone, and this is a work on the right-hand side, work done on humans. But can this be performed in humans in a low-field MRI such that it will be affordable? Currently, MRI systems are very costly, with a price tag of around $3 million and with many additional running costs. My team, and later on, the spin-off company Agile MRI, are working on a whole-body, low-field MRI system for this application, as well as for multiple new applications. With our unique technology, we believe we can lower the price of MRI systems by 2 orders of magnitude. That will make them affordable, the MRI systems affordable and available not only to everybody, but also for many new uses that are currently irrelevant in classical costly MRI system. How it works? It starts with our proprietary algorithms, allowing us to reconstruct MR images with a low and inhomogeneous, that's the critical point here, inhomogeneous magnetic field. This bypasses the need to create high and very accurate magnetic fields that are currently done typically with up to a level of a single ppm. This process is very costly and leads to very bulky systems. This will allow us to create affordable and small MRI magnets for the whole body. The vision from that point is to reduce the need also for local high computing by using powerful cloud computing, and then finally we will be able to deliver images immediately to the doctor's office. This is the path, okay, to this vision of proliferating MRI. We started by building and testing our algorithms for image reconstruction with an inhomogeneous magnetic field, and we know that we are able to do so. Then we built, and you see it here in the middle, we built a small size prototype and tested our algorithms. Currently, we are working on the process of building a full-size prototype to test it on animal. Hopefully, the next stage will be building a clinical product, allowing us to test it, again, on multiple applications and specifically for new uses and new cases that are currently irrelevant for MRI. Thank you so much for your interest.
for Uri?
So Uri, tell us about the competitive landscape here for other mobile MRI systems.
Yeah. Low field MRI became something very attractive for many companies in the past few years. Specifically, we have in Boston, for instance, the Hyperfine company, as well as other companies. And yet, since these companies, what they do is typically create an MRI system with low field, but other than that, with the same technologies that are used for MRI, the price tags of these systems is currently around hundreds of thousands of dollars. And we believe we can drop this by a factor of 10. So currently, we don't know of others that do the same thing with technologies similar to ours.
So cost is a driving example of your technology. How about how good is it compared to other technologies? Comparison in terms of quality of the information derived.
This is a very good question. The main issue is the use of low field MRI. And as you use a lower field, you get to lower signal to noise ratio, as we call it in engineering, meaning, as you said, to images that have a lower quality. Currently, improving the quality of the images is done by various engineering techniques, where on top of them, we're using AI to improve the images. We are able to perform fast images and really remove things like the speckle that we see in low field MRI systems by using AI. Again, this is, of course, in addition to multiple tools that we are using and that other teams use as well in order to improve the signal to noise ratio or improve the interference of electronic radiation around, okay? Importantly, there is an issue of a regulatory process of approving such AI applications for MR images. And the good side, in a sense, is since there is competition, there are already companies that get to the process of regulation of the use of AI to improve images, something that will, of course, help us to do the same things.
Thank you, Uri. Any other questions for Uri? Okay. Uri, thank you. Thank you for taking the time.
Thank you so much.
I just have one concluding remark, and get the slides up. So thank you all. This was our first Colton Symposium at the Yale Innovation Summit. Thank you all to the audience for, I believe, helping make it a success, and obviously thanks to the speakers. And most importantly, thanks to Makoto and his team of Yale associates. These are students, postdoctoral fellows, who are not only doing their scientific work, but also working with the Yale Ventures to advance potential careers in entrepreneurship. So thank you all. If there are questions and follow-up, please contact Makoto. His information is up here. We would also appreciate a follow-up on how this session went. So anticipating we'll do it again next year and how we can make it better and make it more user-friendly and also to get input from you all. So thank you very much.